Today, we will be talking about PCR and STR typing. After our DNA sample has been quantified, the process of PCR and STR typing can begin. As a reminder, PCR stands for polymerase chain reaction, and it is the process of amplifying a targeted region of DNA quickly and accurately in order to obtain a larger sample for testing. Upon getting the quantitative results, we can use a concentration and volume equation to find the volume of neat/original solution we need to dilute 15 microliters in order to get 0.5 nanograms of DNA for testing. In STR typing, the commercial kits are usually produced by one of what's called the Big Three: Applied Biosystems, QIAGEN, and Promega. These are the three chemistries that contain common markers needed for comparison across jurisdictions and are included in all major STR kits. These commercial chemistries are NDIS approved, meaning that they can be used to upload data into different DNA databases. This is a theoretical overview of PCR to show the cyclical nature of the process, starting with separation, then binding of our primers, and ending with elongation or extension of the end of our strand to stop the reaction. Now let's take a look at the components involved in our master mix, starting with our buffer, which will help keep our pH steady, and then looking at our DNTPs, which are the building blocks for replication. Albumin helps stabilize Taq polymerase. Our salts, and in this case will be our Mg2+ ions, help with a wide variety of things, including neutralizing the negative backbone of DNA and stabilizing the complex between primers and template. And lastly, Taq polymerase helps synthesize new DNA strands.
So now that we know everything that goes into a PCR reaction, let's put it all together. Into our strip tube, we're going to add 7.5 microliters of the global Filer master mix that contains all of the components we just talked about, 2.5 microliters of our primer mix, and 15 microliters of our normalized DNA for a total volume of 25 microliters. In 3 other separate wells of our strip tube, we will have an allylic ladder, a positive, and a negative control. We'll take this strip tube and place it in a well plate on the thermocycler for DNA amplification. Let's take a closer look at what happens inside the PCR tube throughout this process. There are 3 main steps in PCR. The first is denaturation. This is where our template DNA is going to separate into 2 separate strands for copying. Now that we have 2 strands of our parent DNA, the next step is annealing. This is where our primers attach to the single-stranded DNA. Now that our primers are attached, our DNA is going to extend. Here, Taq polymerase uses DNTPs to create a complementary strand to the parent strands to give us our full copies of DNA. This process is going to repeat for about 25 cycles, but can be done up to 35 cycles. This is not an unlimited process, as we have only put a limited amount of primers and master mix into the reaction. It is an exponential amplification process, so that we will end up with billions of copies of our original DNA.
Now that you know the theory behind PCR, let's take a look at how it is applied in the laboratory. Once you've made your master mix with the template DNA that you want to amplify, pipette this mixture into your PCR tubes. Use a 96 well plate to hold your tubes during this process. When you have finished preparing your PCR tubes, place your tubes into your thermocycler. The thermocycler is used to control the precise heating and cooling cycles required to perform the different stages of PCR. PCR begins with a stage called initial denaturation. This stage activates the Taq polymerase enzyme and denatures the DNA starting material. The time used for the initial denaturation step varies depending upon the type of Taq polymerase used and the type of starting DNA material. After the initial denaturation stage, the PCR cycle of denaturation, annealing, and extension begins. Denaturation denatures or separates the double-stranded DNA template into single-stranded DNA at a temperature of about 94 degrees Celsius. Next, the annealing stage allows the primers to bind to the new single-stranded DNA, which marks the specific location on the template that will be amplified, and this occurs at a temperature of 55 to 65 degrees Celsius. The last stage in the PCR cycle, extension, occurs at 72 degrees Celsius and allows Taq polymerase to use the primers as a starting point to create a new DNA strand complementary to the template DNA. This cycle will repeat in the thermocycler between 25 to 35 times. After 25 to 35 cycles, Taq polymerase concludes the PCR process by adding an adenosine to the 3' end of the PCR products in a stage called the final extension, and this occurs at a temperature of 72 degrees Celsius.
Now we are going to briefly touch on STR typing. So, what is STR typing? It's a technique that analyzes the number of times a short, repeating DNA sequence appears at specific locations on chromosomes. These sequences are the regions on the DNA strands that are targeted for amplification by PCR, and they are called STR markers, or short tandem repeat markers. The number of repeats present in an STR region is called the allele. These alleles give insight into an individual's genotype only, not their phenotype. It became apparent that we needed a common set of these STR markers for comparison across jurisdictions if necessary, so the FBI developed a set of 13 CODIS markers, coined CODIS core loci, in 1997, which was later expanded in 2017 to 20 total in order to reduce the likelihood of adventitious or random matches occurring, increase international compatibility, and also increase discrimination power, specifically for missing persons cases. But what type of STR markers are actually being looked at on these loci? Specifically, forensics target tetranucleotides, which are repeats of 4 base pairs within these STR regions. These create fewer stutter products, which can typically interfere with interpretation, and it also makes it easier to resolve heterozygotes during capillary electrophoresis. It is also important to know that the primers used in STR typing kits to target these regions during the PCR process are fluorescently labeled. This is because PCR and STR typing is not the last stop for DNA on its journey through a forensic lab. The next step on its journey is capillary electrophoresis.